So thank you for the opportunity to speak here today about neoantigen vaccines for personalized immunotherapy. I'm really honored to be part of this group and potentially make meaningful contributions to patient care at the RTTP. I'll first introduce Mayo Clinic's personalized neoantigen vaccine therapy program, and then I will show the neoantigen profiling results of Shirley and Vanessa's tumors. So what are neoantigens? As shown here, in cells, proteins are degraded and the peptides are transported into ER, where the peptides are loaded onto MHC molecules, and the peptide MHC complex is then transported to the cell surface and presented to the T cells. T cells read each peptide to determine if it is foreign and if it's necessary to mount an immune response. As we know, tumors have many mutations that do not exist in normal tissues. So cancer vaccines are neoantigen vaccines. Neoantigens are mutated peptides from degraded proteins presented by MHC and recognized by the immune system, consequently triggering a adaptive immune response. So the immune checkpoint blockade therapy, or ICB, has revolutionized cancer treatments. So the immune system naturally recognizes neoantigens and responds to cancer. However, for tumors to be clinically apparent, the tumors must have evaded immune surveillance. This is because many tumors express cell surface immune checkpoint proteins and disguise as normal cells. ICBs such as anti-PD-L1 or anti-PD-1 antibodies works by jamming that normal signal so the immune system can see the tumors again. However, the efficacy of ICB remains limited and is well below 20%. Therefore, novel immunotherapy agents are being actively developed, and the neoantigens are one of the most promising next generation agents. So this is a brief overview of how neoantigens work. The tumor cells, like the one in the center, present neoantigens by MHC to T cells. In addition, the antigen-presenting cells, such as the dendritic cells, can also take up and present neoantigens. So together, this process leads to the adaptive immune responses that kill cancer. So this is how endogenous neoantigens work. However, the endogenous neoantigen levels are often really low. So if we could identify what neoantigens a patient's tumor has and supply the extra dose by vaccines, these synthesized neoantigens will train the immune systems to look for tumor. And when ICB removes the tumor surface immune checkpoint signals, the trained immune cells can target the tumor readily. The type of somatic mutations neoantigen come from include single nucleotide changes and small insertions and deletions in genomic DNA. Also, the aberrant splicing isoforms of the RNA transcripts and genomic rearrangements. Not all neoantigens are the same. Some only harbor a single amino acid change, and others, due to protein fusion or translational frame shift, can be very different or entirely new compared to wild-type proteins. The second type are generally more immunogenic. So neoantigens are highly personalized or patient-private. For example, when we studied about 850 breast cancer patients in TCGA, about 99.75% of neoantigens were only observed in either a single patient or less than 1% of the patients. And towards the top, only 18 neoantigens were shared by more than 5% of the 850 patients. Because of this, neoantigen-directed immunotherapy needs to be highly personalized as well. So how do we identify highly personalized and patient-private neoantigens? So as shown towards the bottom of this slide, the neoantigen peptides can be directly identified by mass spectrometry analysis of the MHC binders. However, this process, the proteomics process, can be laborious and requires large amount of tissue. Today, the neoantigen detection is mostly done by bioinformatics analysis of tumor DNA and RNA sequencing data in about 3 steps. So first, the patient-specific MHC is genotyped. And the second, potential neoantigens or neoepitopes from mutated proteins that are expressed in tumor cells are generated. And the last step is the bindings between MHC and the neoepitopes are predicted, and the immunogenicity of the predicted neoantigen is then evaluated. We developed an in-house bioinformatics framework called RealNeo. Here is a diagram of the version 2 of the pipeline. I don't really have time today to describe all details of RealNeo, but we'll highlight one novel functionality of identifying neoantigens arising from aberrant RNA splicings. So as I mentioned, these splicing-based neoantigens contain more than one single amino acid change in the peptide and are potentially highly immunogenic. So the figure to the left is the representative data from 4 different TCGA cancer types: breast, lung adeno, lung squamous, and the liver. So there are 2 categories of splicing neoantigens, one from abnormal splicings due to DNA mutations at splice sites, and the other are from de novo RNA splicings without any DNA splice site mutations. I'll talk about the first category first. So as shown in figure A to the right, we first predicted splicing donor or acceptor sites, gains, and losses. After survey of close to 12,000 cancer cases in TCGA, 13 different profiles of splice site mutations were detected, as shown in figure B. So some of which are frequent and others are rare. The top 7 profiles actually represent about 95% of all splicing mutation events. The simplest profile, as shown in figure C, has a single loss of the original splicing acceptor site happening in about 12.5% of all cancers. This simple profile, however, generated complicated 3 different splicing alterations. Depending on the location of the acceptor loss, either in the middle exon or in the last exon of the gene, it could lead to intron retention or exon skipping. We computed all potential aberrant splicing isoforms from all profiles. Most are much more complicated than this simple example. Next, we kept only the splicings with confirmed expression, as shown by the y-axis in figure D, and are rare by calculating a z-score, as shown by the x-axis of the figure D, using the entire TCGA cohort as the reference. Figure E shows the counts of different splice site mutation profiles in the same 4 cancers. Figure F shows the counts of different types of the splicing products, such as in-frame exon skipping, frame shift exon skipping, in-frame intron retention and the frame shift intron retention. So next, this is how we identified the novel RNA aberrant splicings without DNA splice site mutations. To the right from the top, we started with the junction support reads in the RNA sequencing data. For example, both exon 1 and 4 and exon 1 and 5 junctions were predicted. Next, we used a z-score threshold, assuming these events are rare, and in addition require that the novel transcripts are expressed and the fraction of the novel junctions are at least 10% compared to the canonical junctions. Next, we eliminated E1, E5 when we see evidence supporting E1, E5 junction is actually false positive. So this is followed by another round of transcript quantification and qualification to make sure that the novel splicing events are indeed expressed. The novel transcripts will then be translated into proteins and peptides for neoantigen prediction. I didn't show the data here, but I can tell you that majority of the splicing events are actually de novo. So in the 4 cancer types, as shown in figures A to D, splicing neoantigens are more recurrent in long adeno and long squamous cancers, which is encouraging because recurrent neoantigens will offer opportunities of off-the-shelf, ready-made vaccines. Our computational prediction is fairly accurate. The figure E shows that laboratory validation of the top predicted splicing neoantigens. Some actually bound to MHC stronger than the positive control. So at Mayo Clinic, we have established a personalized cancer neoantigen vaccine therapy program. To date, we have treated several patients through single patient IND applications and have one ongoing phase 1 basket trial for solid tumors. Our first patient is a 25-year-old who has advanced kidney cancers with liver metastases. We identified 21 neoantigens and the patient received neoantigen vaccine plus ICB combination therapy. The patient achieved partial response 4 months after the therapy started, and today, about 8 months later, all tumor lesions in the liver had, on average, shrunk about 60% in size. So today, just this morning actually, this patient started the vaccination of a new batch of neoantigens designed based on the tumor aberrant splicing and the gene fusion products, since our new bioinformatics algorithms are capable of identifying neoantigens from novel or different mutational sources now. So here are some additional data from this patient showing laboratory and clinical validation of the predicted neoantigens. So figure B shows a 93% laboratory validation rate using T2 binding assays for the 21 neoantigens predicted for the first vaccine design. The figure C shows the scan results of multiple liver lesions. Day 650 is when the neoantigen vaccine and ICB combination therapy started. And you can see that by day 920, there were significant shrinkage of the tumors. Figure D shows positive 12-week T-cell activities by ELISpot assays for each of the 4 pools of the neoantigen peptides that constitute the first vaccine design. And figure E is the H&E staining of the tumor biopsy obtained on day 920, which shows extensive necrosis of the tumor, as well as immune cell infiltrations. The last figure, figure F, shows that the protein processing and the presentation probability of the predicted neoantigens are much better than the random peptides. This is the clinical protocol of the ongoing phase 1 trial. So I will skip all the details, but I want to mention that the pain point for us and for patients is the 4 to 6 months long wait for the vaccines. The biopsy, the sequencing, the bioinformatics prediction of the neoantigens, and the vaccine manufacturing and the QC, including sterility tests. So some trial participants unfortunately progressed and became ineligible or passed away before the vaccines and ICV combination could be delivered. So going forward, we'd like to switch from peptides to mRNA vaccines, hoping for faster turnaround, higher concentration of the vaccines, and a lower cost. We're also looking into different delivery mechanisms of the vaccines, including dendritic cells, bispecific antibodies, and TCR-T, instead of just the peptide vaccines. So Mayo Clinic Cancer Center just formed a data science team to support cancer cellular therapies, and I lead that team. So for the next few slides, I will talk about Shirley and Vanessa's case. So we got exome sequencing data from 3 tumors from 2013, 2015, and 2017, as well as a germline blood exome sequencing data from 2021 for Shirley. However, there were only 2013 and 2017 RNA-seq data from the tumors available. So 2015 tumor, we could not predict any neoantigens. So all tissues are FFPE. Therefore, we also did some FFPE variant and noise filtering. And we also removed in-house blacklisted genes from the neoantigen vaccine candidates. I want to make a note that we actually prefer fresh frozen tissues, like any other genomic studies, also for neoantigen discovery. At Mayo, we actually have a next to the OR or biopsy day protocol to ensure that the tissue obtained from the patient goes into liquid nitrogen right away. So I won't go through the process of neoantigen identification and nomination, but total, we identified 4 neoantigens from Shirley's tumor. So the first one is 2 overlapping peptides from TP53, and the second one are 5 overlapping neoantigens from MACC1 gene. Actually, it turns out that MACC1 mutation was very immunogenic, and the neoantigen number 3 is also from this gene, and we identified 4 overlapping neoepitopes. Number 4 is also from MACC1, actually from 11 peptides. So this actually gives us higher confidence of the neoantigen potential for this mutation. So for Vanessa's tumor, we got 3 time points from 2018 and 2 from 2021. And we identified 9 neoantigens as listed in this table. Some mutations actually lead to both class 1 and class 2 neoantigens, and others only either class 1 or class 2. So these would have been a good neoantigen vaccine candidates for Vanessa's treatment. So I'd like to thank my colleagues and the funding we received from Mayo Clinic. I've given several of these talks, and I try to highlight different team members. And today I'd like to highlight and give special thanks to our wonderful clinical study coordinators, Lauren and Kayla, the two names towards the end of the slide. Also recently, Dr. Eric Jessen started to work with RTTP on data from more recent patients. And thank you.